Okay, I'm going to get started. Again, I put information on the board that is useful, including what the music is that I play. Those of you who get here earlier get to hear it because it's not so noisy. I even put up things that people request. Anyway, today I have office hours, 9 to 10, 2084, and we're getting a lot of people coming to office hours, and 2084 is a small room, and if it continues to have more people, we will try to use one of the classrooms if it's available that's near 2084. So be aware if you come and we're not in 2084, you come a little late, look for us in some of the classrooms at the same end of the building. No office hours on Thursday. And here's what I'm going to be talking about today. I looked at the end of the last lecture on the webcast, and I was extremely sloppy in what I said. So I'm going to clarify what I said at the end of the last lecture. Okay, the last lecture I was talking about cell components, and I talked about the plasma membrane a bit, and then the nucleus a bit, and at the end of the hour, I started to talk about ribosomes. And I said something that wasn't quite true. I said ribosomes are synthesized in the nucleus. That's not true. So I want to clarify that because we're going to talk about ribosomes in some detail today. So here's a cell, and here's a nucleus. And there is a region in the nucleus called the nucleolus where primarily RNA is synthesized. There are several kinds of RNA. That's not important for our discussion. It's important for your next series of lectures. What happens is the RNA that's made in the nucleus, the nucleolus region, combines with protein that is moved into the nucleus from the cytosol of the cell to make RNA protein complexes. RNA protein complexes. And the ones we are mostly interested in are the large and the small subunit of the ribosome. The ribosome has two parts to it, called a large subunit and a small subunit. Those are what are synthesized in the nucleus. The ribosomal subunits are then exported out into the cytosol of the cell. And they stay as individual subunits, individual components, until they become involved in protein synthesis, at which point they combine to form a functional ribosome. So a functional ribosome only exists in the cytosol of the cell after the combination of a large subunit and a small subunit. This is very complicated. This is beyond the scope of my lectures, but I need you to know what ribosomes are because we're going to be talking about them today. And off and on, we may refer to them again. So this is sort of how they come about. They are RNA protein complexes. And I said they are very large things. And this is an example. This is the data that exists for ribosomal composition. You can see the eukaryotic ribosome is larger than the prokaryotic ribosome. But both are very, very large complexes. The large subunit, and these are numbers, you don't have to memorize these numbers. I'm giving you these just so you will appreciate how big these complexes are. 3 RNAs, 49 different proteins in the large subunit of the eukaryotic ribosome. 1 RNA, 33 proteins in the small subunit. The composition of a prokaryotic ribosome is different. So they're not identical. The two ribosomes are not identical in prokaryotes or eukaryotes, but they're functionally the same. That is, they are the machine, the factory that makes proteins, even though they have different compositions. An interesting feature of ribosomes is that these ribosomes in prokaryotes, their activity is inhibited by antibiotics, okay, such as streptomycin. Well, I'll write tetracycline first. Tetracycline, streptomycin, not penicillin. Penicillin does something else. So if you go to the doctor and you're not feeling very well and he says you've got a bacterial infection, what he's going to give you are antibiotics that will specifically inhibit prokaryotic ribosomes and kill the bacteria and not disturb or affect eukaryotic ribosomes. This is why the target of these antibiotics are the prokaryotic ribosomes. When you look out in the cytosol of the cell, well, when we talk about ribosomes, we talk about two different varieties, I guess I'd say, varieties. There are what we call free ribosomes. Free ribosomes are in the cytosol of the cell. Then there are other ribosomes which are known as bound ribosomes. Bound ribosomes are bound to membranes within the cell. I'm going to first talk about free ribosomes and what they do, and then I'm going to talk about bound ribosomes. I'm going to skip figure 42 for a little while, come back to it later, but I'm going to refer you to figure 44, which is actually one of my favorite figures. It's a very simple figure, but there's a lot of information in it. Boy, oh boy, I am just twisted up today. All twisted up. There. Okay. Figure 44 shows ribosomes, little box ribosomes, and then it shows you the cytosolic ribosomes, and then it shows you the products, where the products of those cytosolic ribosomes, cytosolic means free, where those products go. They go to mitochondria, they go to the nucleus, they go to peroxisomes. You can add another box, which would be chloroplasts. Those four organelles, the proteins in those organelles are made on free ribosomes and somehow are directed to those organelles by protein targeting, which we will talk about later this morning. The other ribosomes, the bound ribosomes, direct proteins to different parts of the cell. And I will come back to that in a minute. It's going to get a little confusing now, or hairy. What's shown in figure 45 is a complicated figure. There are two pathways for the movement of proteins from these ribosomes. One is called co-translational, and one is called post-translational. Now, what's shown in figure 45, in the middle of that figure, is you've got a large subunit and a small subunit coming together, and the reason they come together is they interact with an RNA molecule, messenger RNA, which has information for making a protein, and the ribosome reads that information. So that's what's shown in the little box above the bigger boxes on the bottom. You can see the ribosome is moving along this messenger RNA, a protein is being made, and it shows you newly formed polypeptide is at the end of that row. So all protein synthesis starts in the cytosol of the cell, okay, out here. But what happens are two different things. We have something which is known as post-translation. What is translation? Well, translation is the conversion of the information in RNA into protein. That's all you have to know. So something is happening after the protein is synthesized. And what's happening in post-translational import is the protein is synthesized, it's released into the cytosol of the cell. That's what's shown at the bottom figure to the right. It shows several things can happen to those proteins. One very important one is they remain in the cytosol. Lots of proteins are in the cytosol of the cell, this liquid medium. But the other things that happen are they can be transported into organelles, such as the nucleus, the mitochondrion, the chloroplast, and the peroxisome. So we'll talk about that first. What was discovered at some point was that proteins which are being synthesized have at their N-terminal end something that is called a signal or transit sequence. there's a relatively small number of amino acids which are found at the end of a protein. This is a large protein, and it may be 10 or 20 amino acids, and somehow these amino acids are involved in targeting the protein to its final destination, be it one of these 4 places that I just mentioned. In some cases, unfortunately, life isn't as easy as we would like it to be. This signal sequence is at the C-terminal end, and sometimes it's actually internal in the protein. But for our purposes, I want you to think of the signal sequence as a region, as a series of amino acids at the N-terminal that allows this protein to go to A, B, C, or D, the four destinations of proteins made on free ribosomes in the cytosol. I've given you in figure 46 some actual examples of these signal sequences. They are meaningless to look at, okay? There's no information in these sequences other than you should be able to see that they're different. Import into mitochondria is different from import into the nucleus, which is different from import into a peroxisome and or a chloroplast. Somehow this signal sequence can interact with a receptor on the membrane of these various organelles. There's a recognition, and then the protein is brought through the membrane into that particular organelle. What happens to this sequence after it's in the organelle is it's just chopped off. So you won't see it in the final functional form of this protein. Is this okay? It's neat. I mean, it's neat because it answers this question of how do proteins get targeted to the organelles that they have to go into, and there is information in these short sequences of amino acids at the N-terminal that allow this to happen. Now, in 3 of these cases, that is the chloroplast, the mitochondrion, and the peroxisome, this is the only thing that happens. The signal sequence interacts with a receptor on the membrane, and the protein is moved into the organelle. In the case of the nucleus, something is different, and that is shown in figures 47. 47 I'll just refer to. It's not relevant to this, but 48 is relevant. Figure 47 shows you these pores, these large pores that are in the nuclear envelope, the membrane around the nucleus. And I think you should be able to appreciate now, based on the size of these ribosomal subunits, okay, these 2 subunits, which are very large, that these things have to go out through a fairly substantial pore, large pores. So large pores are used for transporting things out, big things out. But when proteins are transported into the nucleus, there's a slight variation on the theme of how they get in. There is a nuclear signal sequence, yes, on these proteins, but there is another protein involved. Shown in figure 48 is you see a protein, okay? Off to the left, it has a nuclear localization signal, it's called, for the nucleus, but it reacts with another protein. So the nuclear proteins will interact with a protein known as important. That doesn't mean it's imported. It doesn't mean it's important. It means it's a protein that allows this synthesized protein to be imported into the nucleus. The protein plus important are imported into the nucleus, and then there's some biochemistry that occurs that is not very important to us. Obviously, it's important to the cell. And that protein is released in the nucleus. The important molecule is released in the nucleus, and that goes back out. So protein-mediated transport occurs in the case of the nucleus. Okay, that is post-translational import. Post-translational import. It's post-translational because everything I talked about is occurring after the protein is fully synthesized. Protein is fully synthesized, released into the cytosol, and then all these things happen. If you look at the left-hand part of figure 45, there is another process which is known as co-translational import. And this involves a whole nother set of components in the cell. There is a membrane system within cells which is known as the endomembrane system. I put this on the board because it's important. This system of membranes, the components in it are related to each other by, number one, direct physical contact. If you look now at figure 42, you can see to the right that the nuclear envelope, the endoplasmic reticulum are continuous membranes. So there's a continuous membrane system that runs through the eukaryotic cell, and that is part of the endomembrane system. In addition, there are components that move from one membrane system to another membrane system. For example, there's a transfer of material from the rough ER, and we'll go through this again, to the Golgi complex, to, say, lysosomes, which are organelles. And this transfer of material occurs via vesicles. What are vesicles? Vesicles are membrane-bound sacs. So you have a way of putting proteins into sacs and moving them from one membrane region to a different organelle to a different organelle. The components that are involved in this overall system are the nuclear envelope, the rough and smooth endoplasmic reticulum, ER is endoplasmic reticulum, the Golgi complex, the lysosome, the plasma membrane, and also vacuoles. The initial targeting of proteins along this pathway, as I said, involves co-translational import, shown to the left of figure 45. Now, what's happening here is the protein synthesis starts. It starts just the way the one on the top, the one on the right starts. The protein is being made, but as that protein is being made, the protein is dragged to a membrane. It's dragged to a membrane because the protein reacts with a signal recognition particle. Signal recognition particle. And this is shown on figure 49, the signal hypothesis it's called. I don't know why exactly that. So what happens is you start making the protein. There's a signal sequence on that protein. It's shown in the sequences in 46. It says import into ER. There's a signal sequence. That signal sequence, however, allows the protein to react with a particle. That particle drags the whole protein plus ribosome to the ER membrane, and that's what's shown in figure 49. So what you have on the ER membranes are ribosomes. Those ribosomes are synthesizing a protein, but as the protein is being synthesized, it's being transported into this space which is known as the lumen in the rough ER portion of the membrane. Rough ER has an internal space. Known as a lumen. You'll see this term often in cell biology. A lumen is an internal space of a membrane. And then the signal particle is released, the ribosome is released, and the protein is now inside the rough endoplasmic reticulum. So it's called co-translation because the protein is being imported into this organelle as it's actually being synthesized. It's not released into the cytosol in the case of co-translational import. The free ribosomes, which are floating around in the cytosol, are free. The bound ribosomes, now I've told you, the bound ribosomes, you will find them on the rough ER membrane. They don't stay there. There's a population of ribosomes that come onto the membrane. When they're finished synthesizing proteins, they're released from the membrane. They go back. Next time, they can be a free ribosome. Just the way the large and small subunits of the ribosome dissociate when a protein is finished synthesis, and this small subunit can join with another large subunit the next time around. What drives that, all of that, is getting coordinated on a messenger RNA. So bound ribosomes are identical in composition, identical in chemistry to free ribosomes. It's just that they're on a membrane. And they're on the membrane because they're dragged there because of the signal recognition particle which interacts with the protein being synthesized. So this is a little more complicated. And I'm afraid it gets more complicated. There is a, let me before I continue with the protein story. There's another region of this endoplasmic reticulum. If you look again at figure 42, to the right are actual electron micrographs of those membranes. And you can see in that figure there are little knobs stuck on all of these membranes. Most of these membranes to the right, those are the bound ribosomes. But if you look to the left, it says smooth ER. There are membranes which are known as the smooth endoplasmic reticulum, which do not have ribosomes on them. Well, that immediately should tell you those membranes are not involved in protein synthesis. Well, what do they do? They are involved in synthesizing lipids. A lot of the lipids in a cell that end up in membranes are synthesized in the smooth ER. They synthesize polysaccharides, so they're primarily synthetic, although they also are involved, the smooth ER membranes are involved in what we call detoxification, the breakdown of bad things that get into the cell. So they're doing sort of a general metabolism, not involving the synthesis of proteins. The protein synthesis is occurring in the rough ER membranes. After proteins are made in the rough ER, there's a series of reactions that starts to occur. I've got to erase something. I think we're done with this. So because these proteins that are made in the rough ER have several destinations they can go to. They can go to the lysosome, they can go to the vacuole, they can go to the plasma membrane, or actually they can be secreted from the cell. So again, there's got to be some targeting there. So we have proteins which are made in the lumen of the rough ER. The first thing that happens to these proteins is they're modified. And they are modified by the addition of sugar groups. Shown in figure 50 under the signal hypothesis is the structure of something that has 13 sugar groups. You see a protein, you see all those alpha helices, that's just a generic protein made in the lumen of the rough ER, to which is attached this large sort of flag, if I can call it a flag, this large thing that has 13 different sugar groups. It contains glucose, it contains mannose, it contains N-acetylglucosamine, something we didn't talk about. So you now have, you've gone from a protein like this to a protein like this. 13 sugars on it. Those modified proteins are transported from the rough ER section of the membrane to the Golgi complex. That's the next player in the game. So these modified proteins are transferred to the Golgi complex, which is actually Golgi complex, like a post office. The Golgi complex can sort out different proteins depending on where their final destination is. great, it's like the Golgi complex is, the best way to think of it is, it's like your post office. You put 94707 on a letter. That means it's going to come to around where I live. You put 94720 on a letter, that's going to send it to the University of California at Berkeley. So the post office can sort out these zip codes. Well, the Golgi complex can sort out these zip codes too. And the zip code in this case is not a number, but it's this sugar thing. What happens in the Golgi complex is shown in figure 51, and the details are beyond all of us, I'd say. But there are further modifications of the sugar group. So it comes in, all the proteins that come into the Golgi complex have this kind of 13 residue sugar thing on it. But then some of them are removed in the Golgi complex, or they're modified, or they become phosphorylated. Phosphates are added to them. So that as the proteins move through the Golgi complex is a series of, usually drawn like this, of membranes which are closely associated with each other. Proteins come in, move here, and then are finally sorted out. It's like a plate of stacked dishes. As the proteins move through, they're modified, and finally, at the end of the day, they're sorted. And sorting is shown schematically in figure 52A. You can see the proteins come in. They all have this little black flag on them. Okay, but then they move to different regions of the membrane where there may be receptors there that can recognize one modification versus another. For example, maybe you remove the glucose residues. Well, if you remove the glucose residues, that's a sign for those proteins to be secreted. If you phosphorylate these sugars, or let's say sugars, this is a sign, it apparently is a sign, that these proteins should be directed to the lysosome. The movement of these proteins, as I said, is in bags. They are put into membrane-bound sacs. That's what's being shown in, I think, nicely in figure 52A. So the proteins move to an area, and then the membrane basically forms a little sac around them and comes off as like a bubble. Like you have a big bubble and you can blow a small bubble off it. That's what these vesicles are. And they have specific groups of proteins that are going either to the lysosome, the plasma membrane, vacuoles, and also, I should say, secretion. Lots of proteins are secreted from cells. Certain types of cells are very, very active in secretion. So what you have to become aware of, familiar with, is the difference between this process and this process. The second one is fairly complicated. It involves modification by sugar addition. And there's a term for this. We have a term for everything. Glycosylation. Glycosylation is the addition of sugars to proteins. So protein molecules entering the rough ER space are becoming glycosylated, and then it's the sugar group that is actually targeting these proteins to different places. So it's a little different than post-translational import, where it's the sequence at the N-terminal end that is directing these proteins to various places. Okay, let me say a couple of words about some of these other organelles that I've mentioned. Like I've mentioned peroxisomes, I've mentioned lysosomes, I've mentioned chloroplasts, I've mentioned mitochondria. Since we're talking about the co-translational import system, I'll talk about lysosomes. Lysosomes are unusual organelles. They have a membrane around them. Okay, we know that. they get their proteins. Their proteins come from the Golgi complex, so they're in this endomembrane system. They are very acidic. If you look at figure 53, it shows that circle is a lysosome. And the internal pH of a lysosome is about 4 or 5. Very acidic environment. And the proteins that are in the lysosome are basically digestive proteins. They break down all kinds of molecules like nucleic acids, proteins, carbohydrates, lipids, all kinds of molecules. The lysosome is a degradative body, and it's a hydrolase. These enzymes are all hydrolases, which means they add water across a bond, making two different things. It's the reversal of the sort of condensation reaction. What's interesting about a lysosome is, the first question is why do you do it this way? If these proteins were in the cytosol of the cell, as opposed to being in an organelle, they would obviously break down all the proteins in the cytosol, right? So what you're doing is you're putting them in a bag, you're putting them in a special bag, and they can function at pH 5, but they will not function at pH 7, which is roughly, we'll say for the sake of argument, the pH of the cytosol of the cell. So if a lysosome is broken apart, those enzymes are inactive in the neutral pH of the cell, and that's the protection that the cell has for preventing bad things from happening. It costs the cell money, essentially, to keep the acidic environment of a lysosome, and that is done using the energy of an ATP molecule, which we'll talk about later. But you keep these hydrolytic enzymes separate from the remainder of the cell, and it's sort of a classic example of why you want to have organelles in a cell, because you can put things in the bag that are going to work together, and in this case, you're separating them physically from the remaining parts of the cell. The other thing I've mentioned, let's see, over here, is another organelle which is involved in removal of dangerous, bad things from the environment. I find it very interesting that eukaryotic cells have several different mechanisms and several different components which are involved in degradative breakdown of bad things, let's say. And lost my place, of course. Okay, the peroxisome. The peroxisome gets its proteins by post-translational import. And the peroxisome is an organelle which uses oxygen. So you'll have a compound, which is a toxic compound again, let's assume, which will react with oxygen to form R plus H2O2. So this compound is bad, and it's removed from the cellular environment by an enzyme, presumably. There's enzymes for all of these things. That utilizes oxygen, and it breaks down this compound, and it produces hydrogen peroxide as a product. That's why it's peroxisome. Now, hydrogen peroxide is not a very nice material either. So you have to get rid of it. And these organelles contain large amounts of an enzyme, which is known as catalase. We'll come back to this enzyme. And its function is to break down hydrogen peroxide. Hydrogen peroxide is bad because it's a very strong oxidizing agent. Very, very strong. It will oxidize biological membranes. It can do all kinds of damage. So what this enzyme does is it just makes water and oxygen. Okay? It breaks it down very, very efficiently. It's a very good enzyme. If you look at photomicrographs of of peroxisomes, sometimes this would be a peroxisome. Sometimes you see there's this sort of grid-like thing in this organelle. And what that is, it's basically this enzyme is present in such high concentrations, it forms these, they're not crystals, but these organized structures, and you can actually see them in pictures of peroxisomes. So peroxisomes and lysosomes are both breaking down molecules. They're both degradative organelles, but peroxisome does it oxidatively, oxidation using molecular oxygen, and the lysosome is breaking down materials just by hydrolyzing bonds in those particular materials. Okay, everybody with me? In the remaining time, I want to talk about two other organelles that we will come back to and spend a great deal of time talking about, because they're very important. There are two organelles which we call energy transducing organelles. Producing organelles. And those are mitochondria, which often gets abbreviated MT, and chloroplasts, CT. They're energy transducing organelles because they are specialized in producing the energy compounds that are needed for metabolism. And there will be 4 lectures on these two organelles. That's how important they are. So they make ATP. For anybody who wants to know what they're doing, they make ATP. Obviously, chloroplasts do it in the light, and mitochondria do it in the dark. They have some interesting features. The mitochondria are found in animals and plants. Plants have mitochondria. Chloroplasts are found only in plant cells. They're higher plants or lower plants. They're not part of the endomembrane system. They get their proteins by post-translational import. When one looks at these two organelles, their structure is unusual, and I'm just going to introduce this to you. If you look at a mitochondria, this is an outer membrane, and there's an internal membrane. Looks something like this. So there are internal membranes within mitochondria. That gives you several different spaces, this space and this space, for example. All of these are very important. If you look at a chloroplast, its membranes are even more complicated. There's an internal membrane, but there's stacks of membranes. Membranes are stacked like that. You will when you do your photosynthesis experiment, there will be some micrographs of chloroplasts, and you can see the stacked membrane regions and the unstacked membrane regions. Again, these membranes have internal spaces, and these are very important. So both the chloroplast and the mitochondria have internal membranes. A more interesting feature in relation to what I've been talking about today is both chloroplasts and mitochondria have ribosomes. Okay? So what does that tell you? What? Well, they can make proteins. It doesn't tell you they do have their own DNA. You're ahead of me. Okay. If they have ribosomes, they have the capacity to make proteins. Okay? Because where you have ribosomes, you have proteins. It turns out that both of these have ribosomes. Both of these have DNA. Both of these have RNA. And this is why, when I talked about the nucleus in the last lecture, I said almost all of the DNA in a eukaryotic cell is in the nucleus. That's because in an animal cell, DNA is found in mitochondria. In a plant cell, DNA is found in chloroplasts. the interesting feature of the presence of DNA and ribosomes in these two organelles, and both of what I say for one is true for the other. If you look at the DNA in either of these organelles, it is circular. Okay, what does that mean? What kind of DNA is circular? Is eukaryotic DNA circular? No, right. Is prokaryotic DNA circular? Yes. This is prokaryotic DNA. It looks like prokaryotic, okay? If you look at the ribosomes, they're still here. I saved the right thing for once. If you look at the ribosomes in a chloroplast and the mitochondria, it turns out they are not this size, they are that size. The ribosomes and the DNA in these two organelles is prokaryotic, or looks prokaryotic. It's not directly prokaryotic, okay? So we've got prokaryotic and prokaryotic. Well, this has led to a lot of speculation as to where these organelles came from. And the most widely accepted theory now is something called the endosymbiotic theory model, I guess, let's say model, for the origin of chloroplasts and mitochondria. And that model proposes that you had at some point a eukaryotic organism, simple eukaryotic organism, doesn't have to be human, I mean a bacterial-like thing or something more complicated than that. But that organism ingested a prokaryote. In the case of ribosomes in plants, it could have ingested some cyanobacteria, a very primitive form of photosynthetic organism. And basically, that ingested material, that ingested organism was not totally degraded, but it retained a lot of its properties as an independent component in these two organisms. Because it's hard to see how you can have prokaryotic components within this particular organelle unless the organelle was a prokaryote. That's basically what I'm saying. Now, and you know, as I said, this is the most widely accepted theory, and most people believe it, that there was this relationship between a eukaryote and a prokaryote. The prokaryote got ingested and retained much of its function, well, some of its function, in terms of its DNA and ribosomes. Now, it turns out over the course of evolution, much of the information in the prokaryotic-like DNA has been lost. So a chloroplast or a mitochondrion cannot reproduce itself fully without the presence of the rest of the cell. That is, the DNA of the cell is still functioning, and that DNA is providing some 80 to maybe 90% of the proteins in a chloroplast and mitochondria. And the coding in the DNA molecule is for 10 to 15% of the proteins in the organelle. There are some interesting situations that arise where, for example, you have a protein that has quaternary structure. You know what that means. It's got different kinds of subunits. Some of the subunits are synthesized in the organelle, and some of the other subunits are synthesized, this would be the cytosol of the cell, are synthesized in the cytosol of the cell. And these have to come in to the chloroplast or mitochondrion to form, say, a functional enzyme. And how these two processes are coordinated is sort of an interesting problem that people are, let me just say, worrying about, trying to figure out, you know, how does this guy know, or this guy out here outside the chloroplast say, no, it has to be transported into the chloroplast to form a functional enzyme. There's communication between inside the organelle and outside the organelle. Let's see. I think that's all I'm going to say. I'm sort of caught up with everything. If so I'm gonna stop here because the next topic is membranes, and that's something I just started on Friday. If you have any questions, you've got a few minutes to come up and ask me things. Maybe I can answer them. If not, come down to office hours.